Hello everyone. In this video we are going to talk about the translation with the much more focus on the tRNA structure. In order to find out the tRNA structure, there are so many other websites that you can see the 3D diagram by just typing the name of the structure that you are looking for. There are some links over here. I'm going to show you every one of those links by opening in a separate browser. So let's have a look at together. Okay, I have clicked on the link and this is the website that you find by clicking over the link. 4V42, that's the name of the protein that you have already asked for. Finding the structure, and the name of the website is like RCSB PDB. PDB is protein database. It's very famous name and abbreviation. So anywhere you will see PDB means protein database, and you will find the structures and the 3D structures. For example, here, as you can read, crystal structure of the ribosome at a 5.5 angstrom resolution. That's the magnification of the angstrom, which is, you will find this measurement at the lowest amount when you are using the electron microscopes. So this is a kind of a structure that you will find, and so cool. And if you can click over here, you will see different things. Like if I click on rotation, so nice, it will start to rotate for you. And disulfide bond, if I click, they show the disulfide bond. And if I click this one or this one, yep, it can show the background in black or white. So cool, isn't it? Oh, I found something really interesting. Guess what this is about? Really? Oh my gosh, you see that? That's the COVID-19 coronavirus, recent virus that have killed maybe more than 3,000 up to now. Anyway, this PDB, as you can see, you will find any type of information, and this is about the COVID-19 coronavirus resources. If I click, there are so many things. Because this is something new, I just found in the PDB database, protein database. So cool. I just wanted to show you as well. Yes, nature of science. This is something very important in your IB exam, and I have heard that they are trying to make more questions every year. And that was something last year, and most of the students were shocked about the nature of science type of question. So don't be scared, and don't lose that part. What does that mean? Nature of science here is related to computer, bioinformatics, and those programs. Developments in scientific research follow improvement in computing, and the use of computers has enabled scientists to make advances in bioinformatics applications, such as locating genes within genomes and identifying conserved sequences. So it's all about the computing programs, bioinformatics, and the website that I just showed you, PDB, protein database, have a lot of things. And can you tell me what is this structure? Yeah, this is a tRNA structure. How cool it is, isn't it? So you can just type the tRNA and you will find the 3D structure. You can rotate, you can find any type of bands between the molecules, and so nice. And this is called bioinformatics. Yeah, you see, bioinformatics also relies on computers to a large extent, as it involves analyzing large amounts of data stored in vast database. I think I have mentioned this so many times. If I go back to my graduate school, definitely I will choose protein rather than DNA. That's so nice, and bioinformatics is super cool. Okay, let's move. So here is the tRNA structure. The thing that you will need to learn on the tRNA structure is that the tRNA is activated by tRNA activating enzyme. What does that mean? That means that, look at this diagram here. The amino acid here is the methionine. If the amino acid as methionine wants to be added to this acceptor part, you remember ACC acceptor. If this methionine wants to be added to this, then there should be an enzyme, and by the presence of that enzyme, this ATP will be added to amino acid. In this enzyme is called amino acid tRNA synthesis. This enzyme is making a tRNA with the amino acid, so that's why we call it amino acid tRNA synthesis. And once the amino acid will have the ATP by the usage of the energy, once the ATP is becoming AMP, it loses two phosphate bonds, which means that the energy will be restored, and this means that it becomes activated, and this amino acid already become activated and is ready to attach to the specific tRNA. So what happens that the activated amino acid methionine has been activated by ATP by the loss of two phosphates, become AMP, already activated, and this activated methionine, you see as a star, have the potential and have the power to attach to this acceptor site, and you can see over here. And by the release of the AMP, the energy who has been already activated, stored in the amino acid and make the amino acid activated, is used, and the amino acid can be attached to the ACC acceptor site. That's all. And here are, what I just explained. tRNA delivers amino acid to the growing polypeptide chain in translation. It picks up nucleic acid by the specific tRNA activating enzyme. And this action needs the usage of the ATP. And we have 20 different types of enzyme for 20 different amino acids. Because each enzyme is acting with a specific amino acid for activating that. In common, we call all of them tRNA activating enzyme, but technically we should have at least 20 tRNA activating enzyme, because each of them should act for a specific, each enzyme should act specifically for each tRNA and amino acids. Okay, so the one that is activating methionine cannot be used for activating like the threonine, for example, or other amino acids. So we have 20 of these enzymes corresponding to 20 amino acids, for which the tRNA molecule has a complementary anticodon. And the energy that will be used for this activation is coming from the ATP. That's what I just explained. Let's move. I think this table has been already explained to you many times, and the first slide I just explained this table as a circle, and it is exactly the same, but you just need to go through the first side and then look at the top side, and then finally look at this. For example, let's say, how do I find methionine code? Or if I know the methionine code, do you know that? Let's go once again. August, I love August. AUG, how do I find it? AUG is coding for what? Then you come first from here, A is here, and then you look for U. Which one is U? Only this one. So you know that the AU should be in this area, okay? Should be in this area. And how do I find G? Then you have to look at here. Okay, so A, U, and for one of these, G. So in this case, the G would be here. So AUG is here, August is here, and then methionine already mentioned. So this is another way of that, the first slide, magical circle, okay, it's the same. That's about the genetic code. The codons are triplet. You can pause the video and read by yourself if you want. And we have 64, and you know that why do we have 64? Because we have 4 nucleotides, and each of them has 4 possibilities, and the maximum possibilities of a combination of 3 codons would be 4, 4, 4. That would be 64. And since we have only 20 amino acids, we have more codons than amino acids, so that's why we have more than 1 codon for 1 amino acid. For example, leucine can be coded by CUU, CUC, CUI, or CUG. That's the reason we have more than our codons are more than the number of amino acids. Okay. And about the starting codon August and in the ending or termination, which you can find it over here. There are 3 stop codons, and UAA, UIT, yeah, remember? That's all. Okay, and again, it's a review. I'm not going to review again. How many times I should explain this part? Oh, no way. Go ahead. I'm just skipping this slide. It's about translation, tRNA coming and going to the site. First go to the new one is going to the A site. The previous one is in P site, and then the combination, and then this bond is happening, and then this moves one step over here, and A go to P, and then in P site, and then again new A comes to A. That's all. Oops. Yeah, there are number 1, you read by yourself, number 2, number 3, and number 4. Pause the video and read by yourself. I'm not going to do that for you. Go ahead. Initiation of translation involves assembly of the components that carry out the process. So, the process of translation can be summarized by the following steps. Here we go. I have explained everything for you. I'm not going to explain again. Pause the video and read by yourself. This is about initiation. This is about elongation. And this is about termination. Cool. Next slide. Initiation. I already told you many times. Initiation. Yep. It starts with the combination by the complex, making the complex of the ribosome, the subunits, small and large subunits, and the presence of the sites like EPA sites and the amino acid tRNA bindings. So this is the initiation. So in this initiation, you can see that this is the starting, and then in the initiation we will have the initiation complex at the end. And then in elongation is the process that one by one tRNA will be added, and again next one, and then you will start to have the growing protein. That's the elongation. I am sure that you have time to pause the video and read all of these. And then termination. The termination is happening when the last codon will end up with what? UGR. Yes, these are the termination codons. Any of them will be here, like this one, UGR, and then instead of amino acid, there will be a release factor coming in. So that's the release factor that makes that to terminate, make it terminated. That's all. And the Translation can occur immediately after transcription in prokaryotes due to the absence of the nuclear membrane. Pause the video and tell me, what do you think is the difference between this cell and this cell? Okay, you pause the video, I trust you. This is a prokaryote because the DNA is circular, and this is a eukaryote because the DNA is not circular. That's the one thing. Second thing, you don't see any nucleus membrane, but you see a nucleus membrane. Another second thing that they are different. Third thing, the mRNA after transcription can be used for protein translation immediately, but here the mRNA should comes out of the nucleus and goes through some spliceosome things, and then they should remove the introns and keep the extron, extremely important one, extron, and make it mRNA ready for translation. So these are the three things that I just explained between these two structures, between these two cells. So one is prokaryote and the other one is eukaryote. Yep, there we go. What is polysome? So simple. Polysome is a structure that consists of multiple ribosomes attached to a single mRNA. Once you have the mRNA, then there are multiple polysomes, multiple ribosomes in the length of the mRNA, like this. So that's the electron micrograph, and you can see the number of the electron micrograph showing the polysome, and you can see a growing amino acids, and each of them would be a ribosome. You see, ribosome. So each of these are ribosomes, and each ribosome has a growing amino acid. This ribosome has this growing amino acid, and this ribosome has this growing amino acid. This ribosome has this growing amino acid. So each ribosome has a growing amino acid as a protein. That's why we call it polysome. So 5 ribosomes reading same RNA sequentially, and the starting codon reach August, and then the DNA, sorry, why did I say DNA? And then the mRNA will be read and will be translated simultaneously sequentially with the ribosomes, which are called polysomes. And another graph. So the arrow indicates the chromosomes, and the polysomes that show the chain, show as the side chains. So this would be like multiple zome or polysomes. Each of them is a ribosome complex that has been attached to an mRNA for transcription, showing growing polysomes on a chromosome from E. coli. What is the purpose of transcription and translation? Oh my gosh, everyone knows, including you. What is the purpose? Pause the video and answer the question. Yes, so many things. Catalysis, muscle contraction, cytoskeleton, blood clotting, membrane transport. There are so many different types of proteins. So the purpose of the transcription and translation is ending with a polypeptide or protein. And we have millions of polypeptides with a different functionality. for tensile strengthening, for transport of nutrients and gases, for cell adhesion, for as hormones, receptors, packing of DNA, immunity, so many things. Okay, there is a very nice video related to how a firefly tail makes light. So cool. I paused the video over here and let's watch the video together.